All right. What am I trying to do this? All right, cool. Right, we've been talking about alkanes. We talked a little bit last week about, just introducing the functional groups as well as reactions of alkanes. And now we're going to talk about cycloalkanes. So, for example, the famous organic chemistry hexagon has 1, 2, 3, 4, 5, 6 carbons. So this would be cyclohexane, right? Hex because there's 6 of them. All right, so what do we think about the square? Cyclobutane, right? So just using those same prefixes that we use to denote number of carbons, the square has 1, 2, 3, 4 carbons, so this would be cyclobutane. All right, the impossible one to draw is the 7-sided ring. And so if it's 7, then yep, it would be a hept. So cycloheptane, 1, 2, 3, 4, 5, 6, 7.
So if you have your shape fully connected like that, it's cyclo?
It's cyclo, it's a ring, yep.
We didn't go over that on Friday, right?
No.
That goes on the homework. Also, why can't you on the
Yeah, I can push back that one on the quiz that's built into Canvas. Yeah, that's fine. I'll reopen that up so you guys can work on that. All right, so now when we have stuff sticking off, so the ring is always going to be our parent chain, okay? So if we have a structure like this, where we have a hexyl group or a hexagon with these 3 carbons sticking off of it, we have the same procedure that we've had where we find the parent chain, and again, the ring is going to be that parent chain, okay? Truth be told, in organic chemistry, there are some circumstances where the ring is not the parent chain. We're not going to cover those, all right? We're just going to cover situations where the ring can be considered the parent chain. So again, we're going to find our parent chain. 1, 2, 3, 4, 5, 6 carbons. So my parent chain is that cyclohexane. Okay. This substituent here, 1, 2, 3 carbons. So what do we call a 3-carbon group? Prop. And then what do we put on the end? Not propane. Ane would be for a parent chain. Propyl. Yep. Propyl. Okay. Now, when it comes to numbering our ring, what's our rule for numbering? What do we want our number for our substituent to be? High or low? Lowest possible number, right? And since it's a ring, there's no like natural starting point like there would be for a chain where you start on one end. So for a ring, we just always number from the carbon that contains the substituent. So no matter where that substituent is located, that automatically becomes carbon 1. Okay? Because if I just picked up this chain and rotated it, for example, this is the exact same structure. It's just rotated a third, 120 degrees, right? So again, wherever that substituent is, we're going to call that carbon 1. So it would be very natural to say that this would be 1 propyl cyclohexane, but that's not quite right, and let me explain here in a second. Right? So that would be how we would normally construct our name. Locant, substituent, parent chain. But because that substituent is always going to go on carbon 1, we don't bother to designate it. Okay? We would just say propyl cyclohexane. Because where is that propyl group going to go? It's always going to go on carbon 1. Right? So we would just say propyl cyclohexane. Okay, so then what if we have a pentagon? Like that. Actually, let me put it over here. Take a second and name this compound for me. All right, 1, 2, 3, 4, 5 carbons. So that's a cyclopentane parent chain. These 2 carbons here, that would be an ethyl group. Okay, and again, since there's only that one substituent, we're not going to bother saying 1-ethyl cyclopentane. We're just going to say ethyl cyclopentane. Okay. All right, so, sorry, just go back. So, ethyl cyclopentane, that's what this one would be. Okay, so now what if I have 2 substituents on there? Now all of a sudden I do have to include the location of those substituents because I got to tell how far apart they are on this ring. All right, so let's do something like this. All right, we're going to go through, we're going to do our same procedure, find the parent chain. So my parent chain is a cyclohexane. We got these 2 1-carbon groups here. So what do we call them when there's 1 carbon? Methyl. Here's a methyl. Here's a methyl. All right, and so how do we indicate that we have 2 of the same group? What do we put in front of it? Di. So this would be dimethyl. Methyl. Okay. But now I can't just leave it like that because I got to indicate that these 2 groups are 2 carbons apart. All right, so what we're going to do is we're going to pick up, again, one of these is going to be on carbon 1. Now, a lot of people wonder, like, oh, do you number counterclockwise or clockwise, right? And it's important to note that these molecules here, I could pick this molecule up and flip it over like a pancake, and this would be the exact same thing, right? So there's no logic of clockwise or counterclockwise, because if I flip it over, it's the exact same compound, right? So instead, the name of the game is the same thing that we've been doing. We want the lowest possible number for that next substituent, right? So you can do it both ways. Go clockwise and counterclockwise, right? So we'll go 1, 2, 3, 4, 5, 6, or the other direction, 1, 2, 3, 4, 5, 6. And so which one of these are we going to pick? The purple, right? So 1,3-dimethyl cyclohexane. And again, we have to designate it because that would be a different molecule than 1,2-dimethyl cyclohexane or 1,4-dimethyl, right? Those are not like, again, the whole point is to be unambiguous when we do our numbering or when we do our naming. Cool. All right, so let's just do one more. All right, so what's my parent chain? The square, the 1, 2, 3, 4, so that would be cyclobutane. Okay. These two carbon groups right here would be ethyl groups. All right. And then when I do my numbering, again, I want them to have the lowest possible numbers. So one of them is going to be on carbon 1. The other one's going to be on carbon 2. So 1,2-diethyl cyclobutane. All right, so with these rings in particular, so let me just, we're going to use cyclohexane to illustrate this. All right. On one of these carbons here, let's just pick this one. How many hydrogens are there? 2, right? Because that carbon's got 2 bonds that are explicitly drawn, so the other 2 must be hydrogens. Okay. Importantly, the way that the ring's kind of designed, those hydrogens are going to be oriented such that one is pointing up and the other one is pointing down. Right? If you take that ring and sort of hold it flat, one of those hydrogens is then pointing up in the air, the other one is pointing down. Okay? And how we illustrate that is using these specific type of bonds. We use a wedge, something like this, all filled in. Okay, so for example, here's one hydrogen. This is a bond that's pointing out of the page. All right, so it's like sticking up in the air when you see a bond on a wedge like that. Okay, again, we call that a wedge. The other type is the opposite, which is a dashed bond. And this is a bond that's pointing into the page. Okay, so we can just draw our bonds as straight lines, or we can draw them as dashes and wedges, and that just has this extra bit of information in there in terms of the exact orientation of where that bond is pointed. All right, and this leads to what's called stereochemistry. Stereochemistry concerns the orientation of bonds in a molecule. Okay, and there are many different types of stereochemistry in organic chemistry. We're going to focus specifically on these rings. And so let's take an illustration of 2 different molecules that have different stereochemistries. All right, so let's say that I have 2 methyl groups on this cyclohexane here. Okay, 2 molecules, but in one of them, both of those methyl groups are sticking out of the page on these wedged bonds. All right, so they're pointed in the same direction. And on my other molecule, I have one of them that's on this wedged bond, but the other one is on a dashed bond. This would be called a pair of stereoisomers. Notice that they're not constitutional isomers like we learned about before. because actually the connectivity of atoms is all the same, right? You have those methyl groups located 2 carbons apart on a cyclohexane ring. So the connectivity of the atoms is the exact same for either of them. The only difference is the orientation of those bonds, right? One of them, they're both pointed in the same direction. The other one, those methyl groups are pointed in opposite directions. Okay? So again, we would call this a pair of stereoisomers. They are distinct molecules, right? If you made one of them, it's not going to magically transform into the other one. There's no way to orient that molecule in space that it would ever look like the other one. They are indeed 2 distinct molecules. Okay? So we have to build this into the name their distinction. So first of all, let's name them. Take a second to name this molecule, and then I'll tell you how to go about the stereochemistry aspect of it, right? Find the parent chain, name your substituents, number it, get all there, and then we'll add this extra little bit on top of that. Okay, so they would both be 1,2-dimethyl cyclohexane, right? You got 2 methyl groups located, one on carbon 1, one on carbon 2, and then that cyclohexane ring, right? So if we use our nomenclature rules, we get the exact same name for both of these molecules. But again, they are distinct molecules. So we're going to build the stereochemistry into the name. Importantly, this one, where the methyl groups are located on the same side, this is what we call the cis isomer. So this particular one would be cis 1,2-dimethyl cyclohexane. And the other one, where they're located on opposite sides, this is the trans isomer. So this would be trans 1,2-dimethyl cyclohexane. All right, and what we're going to see when we start talking about reactions is there are some reactions that are actually stereospecific, meaning they would create one specific isomer, and other ones that aren't. So they would just produce a mixture. It would be a 50-50 sort of composition that you would get here, right? We haven't learned about any of those reactions yet, but that's kind of sort of like to give you a window into where we would go with something like this, right? Again, distinct molecules, and the whole point of organic chemistry is to have the name of the molecule point to one specific molecule. So we have to build that stereochemistry into the name. Okay. So the last thing we're going to learn about with our cyclohexanes is going to revolve around these like chair structures here. So do me a favor, pass these around. These are just some like diagrams that we're going to work on here. And you can, well, let me just show everybody first. Does everybody have one? Did you get one? All right, so it turns out that this shape of a cyclohexane molecule is not actually perfectly flat like a pancake, but it adopts what's called this chair conformation. Okay, so what you have is really each one of these lines you would use together, and then you just have a whole bunch of them there so that you can use on your homework and for practice and stuff like that, okay? So in the 2-semester course, we'd make you all learn how to draw this. I'm gonna pick my battles and just give you guys the diagram to work off of. We just need to know how to use this diagram here, okay? So again, this is actually the shape of a cyclohexane molecule. We call it this chair configuration. Because again, those 6 carbons, they don't actually lie flat in the same plane. They sort of kink themselves up so that all those carbons have that ideal 109.5 bond angle. Right? If you calculated the bond angle of a flat hexagon, it would not be that ideal 109.5. Carbon wants to have that 109.5 bond angle. Okay? So it turns out that the only way to do that is to kink itself into this little chair conformation here. All right? We can see that some of these bonds are pointed straight up or straight down. Okay, so we'll take one of your diagrams and use it just to label everything, all right? So these bonds that are pointed straight up or straight down, these are what are called the axial positions. And the other ones are kind of like sticking out, pointing out, or away from the ring. And these are what are called the equatorial positions. All right, so again, cyclohexane is not flat. It kinks itself into this little chair conformation. When it does that, those bonds are going to have one of 2 orientations. Notice that each carbon has one of each, right? If I focus on this carbon right here, it's got 1 axial bond and 1 equatorial bond. That's true for all carbons in that ring. Okay? Cool. You can notice every other one, that axial position is pointed up, and then the other ones, that axial position is always pointing down. All right, so 3 pointed up, 3 pointed down. Cool. All right, so again, cyclohexane kinks itself into this chair here, but it will undergo a chair flip. Okay, so it's not going to be frozen in that one place. Remember, molecules are always moving, they're always dancing around. And in particular, this chair will flip. Okay? So what we want to sort of note here, and I'm going to get rid of this one just so the screen isn't too terribly busy. Okay? One of the most important skills that we're going to need to do with this diagram here is first to number our carbons from one side of the flip to the other. Okay? So I want everybody to note this and you can really start anywhere, but just for convenience, we're going to say this one all the way on the left here. We're going to call that carbon 1. All right? And importantly, when a chair undergoes a flip, the carbons that used to be pointed up, right? Notice how carbon 1 is sitting at the point of a triangle on the left side. When it undergoes that flip, now it's going to be pointed down. So this right here is that same carbon before and after that flip. Right? So at the, and again, we can sort of follow that it's the same carbon because when the flip over here, we can say it's at a peak, right? It's at the tip of a triangle that's pointed up. And then on the other side, now it's at a valley. It's at the bottom, a triangle that's pointed down in space now. Okay? And to be clear, what we're going to do is number here, and we're not numbering in the same way that we would for naming. We're just doing it to keep track of carbons, to make sure we can tell what's what from one side of our diagram to the other. All right? And as long as you can identify that first carbon, carbon 1, the rest of it's really easy. You're just going to pick a direction, clockwise or counterclockwise, doesn't really matter, and just start going from there. Right? As long as you can identify carbon 1 on both sides, then you can just go around 2, 3, 4, 5, 6, 2, 3, 4, 5, 6. All right. So again, identifying carbon 1. On the one side, it is at a peak at the tip of a triangle. On the other side, it's at a valley, at a bottom of a triangle, right? Cool. So then the other thing that we really want to note about what happens during this flip is let's take a look at this bond in particular. Okay. I'm going to put a methyl group on there just so we can sort of easily see. And that's why you have this diagram sort of with those bonds drawn in. We're going to put a methyl group right here, CH3. That methyl group is what? In the axial or the equatorial position? The axial position, right? It's pointed straight up. Cool. So then when we undergo our flip, that methyl group is going to stay on carbon 1, right? It's not going to jump to a new carbon. That doesn't make any sense. It's the same molecule. It's just undergoing a different orientation, right? But then let's look at what happens to this methyl group. It's on this bond here. Now, that bond is here. And what would we call that position? The axial position or the equatorial position? That's the equatorial position, right? So when we have our flip, our axial becomes equatorial, and vice versa. All the equatorial positions become axial. Now, if you have like a naked cyclohexane ring, they're all hydrogens on there, it will flip back and forth between these conformations and spend an equal amount of time in either one, right? There's an exactly 50-50 distribution of both of these chair flips if they're all hydrogens, okay? But a methyl group is actually like a big, bulky group, right? I mean, we kind of drew it small here, but if I wanted to like explicitly draw that in, that's a carbon, and it's got these 3 hydrogens sticking off of it. right? So it's kind of like this big bulky group on there, okay? So if you put a bulky group on there, a methyl group, or even a methyl group is actually one of the smaller ones that you can put on there, anything large on there, all of a sudden it's going to prefer one of these conformations over the other one, okay? So one of the things that I would ask you is, like, fill in this diagram, make sure you're sort of following along where that methyl group goes. It's axial over here, it's equatorial over here. And then I'm going to ask you which one of these is the most stable conformation, okay? And the rule here is that large groups like to be in the equatorial position, okay? So large groups prefer the equatorial position. Can anybody guess why that might be? More space, exactly, right? So molecules are claustrophobic. They hate being next to other molecules, right? They all like their space. And the axial position is very claustrophobic. There are other axial groups pointed right next to each other, all in the same space. Whereas in the equatorial position, it's kind of pointed off all by itself, right? So this is absolutely just a space argument, what we would call sterics, okay? The axial position has more steric hindrance. Is kind of the fancy science-y phrase. Right? Steric hindrance meaning it's bumping into other atoms. It doesn't like that. Okay? So, from scratch, what type of problem should we be able to do here? Okay? Something like this. So again, I'm just going to make some more room here. Oh, whoa, what did I just do? All right, so we're going to complete the chair flip diagram. For this 1,2-dimethylcyclohexane, is it in the cis or the trans conformation? It's not a conformation, actually. Stereochemistry. Trans, right? Those are pointed on opposite sides, so that's the trans isomer. Okay? So we're going to complete this diagram for this particular molecule here. All right? I want everybody to cover up their first example, their little cheat sheet that they made up at the top, and identify what carbon 1 is on either side of that flip diagram, right? That's going to be the hardest part, the easiest place to make a mistake. So cover up what we just did and see if you can't recall from memory how to identify carbon 1 on either side. Bless him. All right, and let me just say, that is a very important study skill. It took me a long time as a student to realize, but oftentimes we know where in our notes to look, and we rely way too heavily on that. If you always know where to look for a piece of information, that's kind of a signal to your brain that's not really that important to remember, right? And just an example, like, I don't know my mom's phone number, straight up. Like, I couldn't recall it from memory because it's just so easily accessible on my cell phone. I haven't bothered to have to memorize it, right? Back in my day, you memorized all your friends' phone numbers because that was the only place that you could get them, right? If that piece of information is so easily accessible, your brain does not bother sort of storing it in that long-term space. So when you're studying, you want to make sure that you're doing that sort of thing. If you find yourself cheating all the time, you need to sort of like wean yourself off of looking in your notes for that particular answer, right? Studying your notes is a dangerous thing. Studying with your notes closed is kind of like the skill that you need to be at for an exam because, of course, you won't have your notes, right? Cool. So again, identifying carbon 1 is kind of the most basic first step that we need to do. Over here, it's pointed up, and over here, it's pointed down.
Is it
right?
okay if you just showed like the other?
You could have said that this is carbon 1 and this is carbon, oh, that wouldn't be correct though. And this is carbon 1. You just have to be consistent with what you're saying. I think that spotting the one all the way on the left is the easiest one, so that's why I kind of defaulted to that. But no, as long as you're consistent from one side to the other, it's fine. Okay, so then let's put in our methyl group. And for the first one, it doesn't matter which one you pick as long as you're consistent, right? So in this case, I'm going to stick it on this group right here. All right, and just to color code from my original structure, we'll say that this is carbon 1. Okay, so now put in that methyl group on your flip as well, on the right-hand side. Where should it go? All right, so remember, if it's axial on one side, it's got to be equatorial on the other side. So that would be that one there. All right, and then we're just going to pick a direction to number. I'm actually going to go counterclockwise because I think it's just going to be easier to draw it in here. Okay, I have this other methyl group, right? On this carbon here, I'm going to color this carbon blue. Which carbon should that methyl group go on in my flip diagram? Carbon 2, right? They're located 1, 2 apart on my structure up here. So I'm going to need to put in that methyl group on carbon 2 here. Okay, now this is the next trickiest bit here. Okay, in this structure, in my target molecule, again, we said that that was the trans conformation, right? So those bonds are pointed on opposite sides. So I need to make sure when I'm filling in my diagram here that I put it on the right bond. Okay, if I put it on this bond here, this would be the cis conformation because now it's on top over here, right? So again, this would be the cis position, because this one's the top bond over here, and this one's the top bond over here, right? So that would be pointed in the same direction, the cis conformation. So I need to put it on the other bond down here. This is the trans conformation, so that's where my methyl group should go. Yeah, so if we look at these two carbons, we can call this bond up here the top bond, and this bond the bottom bond, right? One's on top, one's on bottom, okay? if I have a trans conformation, these bonds need to be pointed in opposite directions, right? So then when I go over here and I look at this carbon, I can again label that this bond is on top and this bond is on bottom, right? And again, since it's the trans conformation, they need to be on opposite sides. So if I drew one on the top, the other one has to go on the bottom. Yep. It's the trickiest aspect of this whole thing, for sure. Okay? What you cannot do is memorize that, like, trans is always equatorial or something like that. It doesn't work like that. Okay? We'll do another example here in a second where we'll see everything gets messed up. All right? You got to look at that carbon, say, okay, which bond is on top, which bond is on bottom. If this is the trans conformation, I need to make sure that they're in different positions. So then fill in on carbon 2 in your other diagram where that methyl group should go. All right, so on my diagram here, we're going to take a vote. I'm going to call this one A and this one B. Who thinks our methyl group should go on position A? And who thinks our methyl group should go on position B? Excellent, okay. All right, because if it's axial on one side like it is over here, it's got to be equatorial on this side. Right, so then we're going to do all this work, and then we can ask ourselves, which is more stable? And remember, how did we answer this question? Because large groups are going to prefer the equatorial position. So in this one here, what do we think? The left or the right? Which one is going to be the more stable conformation? In this position, or on this left-hand side, which position is this? This is axial. And what about this one? Also axial. All right, we go over here. Which position is this? Equatorial. And which position is this? Also equatorial. Right? So then in terms of which one's going to be more stable, these large groups, they like being in the equatorial position. So this one here is the more stable conformation. Right? What that practically means is if we had the ability to, like, freeze time and then zoom on down into a sample of cyclohexane, we would see like 95% of it in this conformation and only 5% of those molecules in this conformation, right? The more stable conformation is going to be the one that's preferred by those molecules. That's not to say that it's not bouncing around a little bit, but it'll spend more time in one conformation than the other. All right, so let's see how far we can get with another example here. I'm going to do something similar. But now I'm going to move this to be over here. Let's see if I can pick up that blue. Damn, that is not going to work. Okay, so if there was a shortcut, I would teach you the shortcut. There is no shortcut. That's why we have these diagrams, right? So in order to figure out which one's going to be more stable, you have to use the diagram. You have to go through that process, right? That's how we solve these problems. There's not sort of this easy thing where we can remember that cis is always going to be equatorial or something like that, right? We got to use the diagram in order to get at this sort of problem here. So given a structure like this, now we're going to use 1,3 dimethyl, trans 1,3-dimethyl. And you guys are going to fill in on your diagram here, first by identifying that first carbon on either side, then putting in those substituents. Okay? So try it on your own. I'm going to give you guys like a head start, and then just for the interest of time, I'm going to sort of quietly go in and fill it in on my own as well. So if you guys are getting stuck, you can look up and see what's up. All right, so let me just real quick walk through this. So I got my carbon, I got my ring numbered, right? I'm going to put that first methyl group in. You can choose either one. Pick the axial position on carbon 1, right? It would be that you'd be able to get the right answer regardless of which position you chose, okay? So put that in. My next methyl group I need to stick on carbon 3 this time, right? Because that was my structure. So I'm going to put it on this one. And again, this is a tricky part here. This was the trans structure over here, right? Pointed in different directions. So again, I'm going to go to my bonds and I'm going to say, okay, this one's the top and this one's the bottom. Go over to the next carbon. This one's the top and this one's the bottom. So now which one is my CH3 going to go on? The bottom position, right? So CH3. It's got to be pointed in opposite directions because this is the trans structure. So then I do my flip. I'm going to make sure that my axial becomes an equatorial and my equatorial becomes an axial. Okay. So then finally we can get at which one is going to be the more stable conformation. If I look on the left, again, what's this position right here? This one's the axial. And what's this one? This one's equatorial. So then when they flip, this one's equatorial and this one's axial. So is one of these more stable than the other one? No, they're both going to have an axial methyl group and an equatorial methyl group. So these ones would be equal, right? 50-50 distribution if you were to freeze time and go down there and look and see what's up. Okay, so one of your homework assignments is having you practice this exact thing. I will say that it's basically just asks like, oh, is this group axial or equatorial in the most stable conformation? What that means is it's like a 50-50 guess, right? Bottom line is it's really easy to guess your way through that assignment and get full credit. You want to make sure that you know how to use this diagram, right? That's the whole point, and this is what's going to be on the exam. So do it right and use the diagram to answer those questions. Don't just kind of wing it, all right? Cool.